In the last lecture, we've discussed the structures of sugars, especially focusing on the monosaccharides, as well as to understand how they form into oligosaccharides and the polysaccharides. In this lecture, we'll focus more on the polysaccharides, especially covering their structures, functions, as well as their modifications for various biological functions. First, let's talk about the structure and biochemical properties of polysaccharides. First, the structure, they can form homopolysaccharides. It's also called as a homoglycan, where the polysaccharides are consisted of just a single type of monomer or the monosaccharide, just like a glucose. They can also make the heteropolysaccharides, also called as a heteroglycan, where the polysaccharides are consisted of at least two different kinds of monomers. It can be multiple too. And polysaccharides can form either linear or the branched structure too, and depending on their structures, they have different properties and also involved in different functions. Largely, the polysaccharides are involved in these four different kinds of biological functions. Starch and glycogens are involved in storage of the energy source, and they also involved in the structures, and the cellulose and the chitins are the examples for that, and also involved in bacterial cell wall, which is for the structure as well as protection of the cells, and peptidoglycan is a good example for that. And lastly, also play a role in the recognition and the cell surface polysaccharides and some of the sort of a decoration of those sugars on the proteins, will the involved in cell recognition or the cell signaling pathway to control the cell functions as well as in their weight control. So one of the functions of a polysaccharides that we listed was the storage of the energy source. And why would you want to use polysaccharides but not the monosaccharides for the storage? If the monosaccharides are used for storage, that will greatly increase osmotic pressure. And remember that osmotic pressure depends on the total number of solute molecules in water, but not the kind. So regardless of their size of the molecules or the type of molecules, as long as you keep the same number of the molecules, you will have the same osmotic pressure. And that can be explained by the colligative property of water that we talked about early in this course. So imagine that you have 10,000 monosaccharides. Then you're going to have a really high osmotic pressure because of these high number of molecules. But if that is combined to just one big molecule as a polysaccharides, then you can lower this osmotic pressure 1 to 10,000 basically using this macromolecule. So it saves a lot in terms of cells homeostasis. Starch is the most common storage polysaccharide in plants, and you can see it a lot in potatoes as well as corns. And it is a mixture of glucose polymers, so it has the 10 to 30% of alpha amylose, which structure is shown here. It basically has a linear chains of those of thousands of units. The units are the glucose again, which forms the amylose, as you can see here. And the linkages within the alpha amylose is alpha 1, 4. So by this time, you should be able to tell if the linkage was alpha or the beta, and also 1 to 4 or the 1 to 6 and so on. If you are not sure, please go back to the previous lecture and study a little bit more about that. And the starch makes the left-handed helix, as you can see here, as a polymer. And it also contains roughly 70 to 90% of amylopectin, which structure is shown here as well as here too. And this one, the starch basically shows the branched structure where the linkages are alpha 1 to 4. And, but every 24 to 30 residues within this polymer, it'll have the branching. and the linkage will be the alpha 1 to 6, and that goes up to 1,000,000 glucose units. So that makes the starch among the largest polymers in nature. Digestion of the polysaccharides is a sort of a team effort where multiple enzymes and processes are involved, pretty much sequentially. So as a first step, amylase, which is included in saliva, breaks down the starch or the polysaccharides by breaking some of the alpha 1,4 bonds. So they're going to have a little like a chunkier but the smaller pieces of amylose. After that, the pancreatic amylase, which is secreted in the small intestine, break them down to a mixture of alpha 1,4 linked disaccharides, which are the maltoses. They consist of 2-glucose, as well as a trisaccharide, which is maltotriose, and basically contain the 3-glucose as their monomers. And after that, alpha glucosidase in small intestine removed sequentially one residue at a time at alpha 1,4 linkages. And lastly, the debranching enzymes such as isoamylase cut the also alpha 1,6 linkages in amylopectin as well as alpha 1,4 for the amylose too. Glycogen is another storage polysaccharide especially used in animals. And glycogen is prevalent especially in liver, which takes up 10% of its weight, and also in the muscle it has the 1 to 2% of weight as glycogen. And it's similar to amylopectin when you look at the structure, but more highly branched in glycogen. So every 8 to 12 residues have this alpha 1 to 6 linkages, and it can be quickly metabolized when blood sugar gets low. And the fact that it has branched structures, meaning that it has many non-reducing ends, basically of the old ends here of the branches, and that also means that it's easy to mobilize quickly. And the enzymes involved in the breakdown of this glycogen is the glycogen phosphorylase, which cleaves the alpha 1,4 linkages from non-reducing end, and also glycogen debranching enzymes attack the alpha 1 to 6 linkages too. But the most of the energy is stored as fat actually, which take up less weight and less space, so it's more efficient in terms of the energy storage. However, but it is really slow to catabolize. Cellulose, which has a structural role, is the most abundant natural polymer in nature. So it is up to 80% of the dry weight of the plants. And specifically, it is a major component of the plant cell walls, and it confers the rigidity and the mechanical strength. And that property is used for the structural role of cellulose, which gives plants a way to deal with the osmotic pressure. So the plant cell itself has the low osmotic pressure, so if there's no cell wall, then all the water will rush into the plant cell, make it burst out. The cellulose is a linear homopolymer of beta-D-glucose linked by beta 1,4 linkages. So keep in mind that starch, or the amylose, has the alpha 1,4 linkages with the alpha D-glucose, but it's different using the different units, different linkages. And it makes the long linear polymer chain as long as 15,000 units. So it can be really long polymer too. And the chains lie side by side basically to form this microfibril structure and also forms the sheets. And these sheets stack in a way that allows the staggering of the chains just like the bricks in the wall. So it's staggering here like that. And it forms also the cellulose fibrils here in the higher order structure too. And there are 3 types of hydrogen bondings are involved in that cell wall structure or the cellulose structure. Intrastrands, interstrands, as well as intersheet too, which make it very strong and resistant especially to the hydrolysis. It is insoluble too. Chitin, another structural polysaccharide, is similar to cellulose in its structure and functions, and it is used a lot in exoskeletons of the crustacean, spiders, and insects, and so on. So chitin has the extended linear chains made of N-acetylglucosamine. It's also called as glucnac, and gluc was from the glucosamine. The NAC was from the N-acetyl. And it basically has the NHCOCH3 group instead of the hydroxyl group in the cellulose to get this chitin. But it has the same kind of linkages as the beta 1,4 linkages, and it can make the long linear polymer too. And chitin also has the extensive hydrogen bonding similar to cellulose. Glycosaminoglycans or GAGs or mucopolysaccharides are long unbranched polysaccharides consisting of repeating disaccharide units. And these are the examples of glycosaminoglycans where you can see again disaccharides as their units. And this unit consists of an N-acetylhexosamine and a hexose or hexuronic acid, either or both of which can be sulfated too. And the sulfate happens on these yellow pointed regions. And here, the hexuronic acid, unuronic acid means that it's a sugar acid. So it's an acidic molecule but contains the sugar. And a sugar, when you look at it, in which the terminal carbon, the C6 position, which is supposed to have the hydroxyl group, but the dysfunctional group has been oxidized into a carboxylic acid. And the combination of the sulfate group and the carboxylate groups of the uronic acid residues give them a very high density of negative charges. And that means all these huge macromolecules will have negative charges, so it can actually be working as a polar substance, can interacting with the positive charged molecules. And members of the glycosaminoglycan family vary in the type of hexosamine. So these are the example of all different type of glycosaminoglycans. Also, the hexose and the hexuronic acid can be quite variable too. And they also vary in the geometry of the glycosidic linkages too. So it can be here, so like 1,3 linkages, and also 1,4 linkages, and it can be either alpha or the beta too. So which can give rise to the many different kinds of combinations of these disaccharide units, and also it can confer different properties of the glycosaminoglycans. And this slide shows all different type of glycosaminoglycans with the different components. Again, all of them are repeats of disaccharides, but they have slightly different the modifications on their functional groups. Of course, they will have different names with mostly sulfated. Glycosaminoglycans can be found in many different organs, and especially in collagen fibers, in extracellular spaces of connective tissues such as cartilage, tendons, and so on, as well as synovial fluid which lubricates the joints and vitreous humor of the eye. The collagen fibers are embedded in ground substance of glycosaminoglycan, which is typically 250 to 25,000 residues long. And they are slimy and mucus-like, so it gives the property of high viscosity and elasticity of the collagen fibers. And again, since these glycosaminoglycans are negatively charged because of the sulfates, it can easily bind some cations such as potassium, calcium, as well as sodium cations pretty tightly. And it is typically highly hydrated. Again, works as a polar substance, and so it takes up to the 1,000 times volume relative to the non-hydrated form of glycosaminoglycan. And degree of viscosity depends on the degree of shear stress. And it can work as an excellent lubricant and shock absorbers because of this high volumes as well as the elasticity and the viscosity. Glycoproteins are proteins decorated with the sugars, and this sugar play a critical role in regulating such proteins. They're responsible for targeting of the proteins for their subcellular localization, sorting of them in the cell, as well as recognition of the interaction partners, and also the protection of those proteins from their degradation. And these modifications by sugar are not encoded in DNA, so it happens only post-translationally, meaning that there are a lot of variability, which gives the high degree of heterogeneity of glycoproteins and their basically degradation of the sugars. And there are two types of glycoproteins and their linkages to the sugar. One is the O-linked, which structure you can see here. There's a hydroxyl group in the serine and the threonine side chains, and that is actually linked to the sugar and make the O-linked glycoproteins. And the other one is N-linked glycoproteins, where the amide nitrogen of the asparagine side chain is linked to the sugar. Here, this slide shows an examples of O-linked saccharides and N-linked saccharides too, and not only just the monosaccharides, but disaccharides as well as oligosaccharides can be attached to the proteins to make the glycoproteins. And various type of monomers as well as linkages can be used for generating the glycoproteins. So in case of a ribonuclease B, it has a GLUC-NAC and mannose that is attached in just like this linear structure to the asparagine, as opposed to human IgG actually has the branched form with a mixture of GLUC-NAC, mannose, fructose, as well as galactose and so on. And many different kinds of such linkage can be happen. And all these basically makes the proper function of such proteins, as well as sort of like a regulation in terms of their activity, as well as interacting partners control for such proteins. This table lists the oligosaccharides found in the A, B, and H antigens, and this cosylation, which is the reaction to make a glycoprotein, occurs at the non-reducing ends of oligosaccharides. And H antigen is the precursor of A and B antigens. So depending on what type of glycosylation happens on the H antigen, it determines the type of the antigens in erythrocytes. One particular kind of glycoproteins are proteoglycans, which is a family of glycoproteins containing glycosaminoglycans. And we already talked about these polysaccharides, their linear structure, elastic viscous properties, their units, which are the disaccharides, as you can see here as examples, as well as the negative charge of these polysaccharides. And this interesting property actually makes this sort of a stream-like structure, really elaborate structure, with a combination of the sugars and the proteins. And an example of these proteoglycans are these polysaccharides with the collagen in the cartilage joints. Again, the proteoglycans make this interesting higher order structure. And when you look at this structure more closely, there is a hyaluronic acid polymer backbone that goes through, make this kind of string. And also there are bristles, which are basically consists of the core protein, as well as the glycosaminoglycans that are linked to those core proteins. So overall, it looks like a bottle brush. And they are very much highly hydrated because of the property of the glycosaminoglycans that are attached to the core proteins. And again, there are lots of glycosaminoglycans, so there are a lot of water molecules that can be contained here. And also it has a property of a high resilience. So when the pressure is applied to these structure, then it can squeeze out the water away from this structure. But the rehydration happens when the pressure goes away, so it's released. And it is responsible for nourishment of cartilage in joints too, where there's no blood vessels, because you can basically put a lot of water molecules there and contain them. Another interesting and important type of glycoprotein is peptidoglycan, which makes the bacterial cell walls that are required for protection in hypertonic medium to fight against the osmotic pressure, and is responsible for virulence. These peptidoglycans are polymer of a beta-1,4-linked GLUC-NAC and NAMA disaccharide unit, where the GLUC-NAC is the N-acetylglucosamine and NAMA is the N-acetylmuramic acid. And this is also linked to a tetrapeptide, which consists of 4 different amino acids. And in bacteria, largely have 2 different types, gram-positive and gram-negative. And the most prominent difference between these two is that gram-negative bacteria contain outer membrane, which also covers the peptidoglycan. But both type of bacteria do have a peptidoglycan to make the cell wall, and use the same disaccharide units as well as the same structures to protect these cells or the bacteria inside of the cell wall. So using this GLUC-NAMA disaccharides, a linear chain of polysaccharide can be formed, which is also attached to these peptide chains. And the peptidoglycans can have a very rigid structure by cross-linking these linear chains. So this is an example of the peptidoglycan in SRS. And as you can see, this is GLUC-NAC and NAMA disaccharide units that forms this linear structure. And these 4 beads are basically the peptide chains or the tetrapeptide, where you can see its actual structure here. And also these strings or the linear chains can make the high cross-linking by the penaglycine bridge, which is a consecutive 5 glycines. Penicillin, which you can see its structure here, is a well-known antifungal reagent that inhibits cross-linking of peptidoglycans. So it prevents the bacteria from synthesizing new cell walls. So how it really works is that this penicillin specifically binds to the beta-lactam ring of the DD transpeptidase. And DD transpeptidase is a critical enzyme to make the cross-linking between the tetrapeptides or the peptide chains of the peptidoglycans to make these rigid structures. So without this active DD transpeptidase, there's not going to be any cross-linking happens, so that the cell wall is going to be degraded. So these are the monosaccharides and polysaccharides whose structures you should master and memorize too. So here are just a couple examples for the linear structure, glucose and lactose, for cyclic forms for them too, as well as GLUC-NAC, and also for the polysaccharides, cellulose, chitin, starch, and so on. And also you should definitely understand the glycoproteins, as well as different type of glycoproteins and their usages and functions and how they are synthesized too.